Awesome. Thank you, Margie. And thank you all for being here this evening to hear this talk. So like Margie said, this is research that was done for my master's thesis. So if you came to my events, this is going to sound a little familiar today, but a little different. So the title of the talk tonight is The Secrets That Feathers Hold, Using Feathers to Trace Migration. So before we really get into all of this, I just wanted to take a brief moment to really appreciate the amazingness that are feathers. Feathers are these incredible structures that allow birds to fly, to stay warm, to camouflage, to sometimes attract a mate. But we also can use feathers to learn about the birds. So we use feathers in research. During the banding process, we can use a feather to understand what species it is. We can use that feather to understand age and even sometimes sex of the bird. So feathers are really these incredible structures. And while I'm not going to be talking a whole lot necessarily directly about feathers, I'm going to be telling you about how I used feathers to answer some questions about migration. So the techniques that I'm going to be talking about today, I'm going to be talking about in relation to raptors, but these techniques can also be applied to many other different systems, as well as many other different types of birds. So here on the screen are some of the many raptors that we could potentially see here in Minnesota. And I'm just going to be talking about 3 of those species today. So like many other birds, many raptors are migratory. So what this means is that they inhabit multiple different locations during a full cycle of the year. And animal migration is mostly driven by resources. So an animal is likely to migrate from a region of low or decreasing resources to an area of high or increasing resources. And the 2 main resources that drive bird migration are, number 1, food, and number 2, nesting habitat. So for example, here in the Northern Hemisphere, in the fall, birds leave their breeding grounds in the north and move usually south to areas with higher food resources. And then when it's time for the next breeding season, migrants head north towards regions that have more opportunities for nesting habitat, as well as increasing resources during the spring and summer months in terms of food. So you can see in this animation, I love this animation from Cornell, that already you can see that birds have kind of different migration strategies. So some of these dots, which each dot represents a different species, some of them are moving very far, some of them are moving in that north-south direction, but others are maybe only moving locally or up and down in, we can't necessarily see altitude, but they could be doing that. And they might even be moving in kind of an east-west direction. So here in northern Minnesota at Hawk Ridge, raptors tend to concentrate along the western edge of Lake Superior at Hawk Ridge due to a variety of landscape features. So raptors tend to follow the boreal forest, which is shown here in this green color. And they follow this suitable habitat down towards northern Minnesota. And then many raptors tend to avoid crossing large bodies of water due to the kind of this lack of thermal air rise over water. And that means that raptors reach the shore of Lake Superior and they tend to funnel down through Duluth. And then kind of the third and landscape feature is the basalt ridges that line the coast of Lake Superior. And what these do is provide an updraft for raptors that makes flight less energy consumptive. And what this results in is a pretty impressive visible migration in the fall that can be viewed at Hawk Ridge. And so I hope that many of you have gotten a chance to view this. It can look like this or with many other birds in the sky on a beautiful fall day. So if you have quick access at the moment to maybe the chat or to some reactions, you can tell me if you've ever spent a beautiful fall day up at Hawk Ridge viewing this migration. It's pretty spectacular. So why do we even care about raptors? Why should we study them? And why are there many different places across North America that do study them? Well, there's a variety of different reasons, but one really great reason is that raptors are incredible bioindicators of ecosystem health. So first of all, raptors are this very diverse group of birds. So you can see in this phylogenetic tree here that we have a variety of different species that are considered raptors. And those span from things like falcons and caracaras to things like hawks, eagles, and owls. And what this means is that these diverse species also inhabit a wide variety of ecosystem types. And so if we monitor all of these species, we might be able to tell what's happening in a wide variety of different ecosystems. Another reason why they're great bioindicators is that they are top-level consumers. So they're predators, and therefore they're eating at a high trophic level. And what this means is that they are really kind of surveying the landscape and are affected by many different things that are going on within their environment. And what this can lead to is that they are particularly sensitive to human disturbances and environmental contaminants. So contaminants have this ability to bioaccumulate in the tissues of an individual over prolonged exposure, which is shown here with these fish. Over time, they're getting more of this contaminant in their tissue. And then within a food web, contaminants may biomagnify up trophic levels, causing raptors such as this eagle to have a higher dose of a contaminant compared to a lower level. And probably the most well-known example of this is the decline of eagles in response to DDT. So it was kind of long-term raptor monitoring that actually gave evidence to this decline and led to the eventual banning of DDT in the US. So this is pretty incredible that raptors have this amazing potential to actually enact conservation of ecosystems as a whole, just by monitoring them. So at Hawk Ridge, consistent monitoring of raptors has gone on since 1972. Before that, some sparse efforts were done, but in 1972, everything became just a little bit more consistent. So each fall, over 60,000 raptors are counted and over 2,500 raptors are banded. And this represents 50 years of monitoring that has led to the creation of a lot of data that has shown us patterns and leads to even more questions than we have answers to. And one of the pieces of information that we really don't know a lot about is where are the raptors migrating from in the fall? So generally, we assume that most are migrating from north of Duluth, but how far and with what patterns they move through Duluth, we don't really know very much about. So the two main objectives of my thesis work were to estimate the natal origins, meaning the places that the raptors were born, and to determine whether there were migratory patterns in relation to those natal origins. So since I had these objectives, I decided to implement a stable isotope approach to understand where these birds were coming from. But before I really get into that, I just wanted to say that there are many different ways to study movement and where birds come from and where they go. And we tend to think about those as being extrinsic techniques, so things that you attach to a bird to learn about their movements, and then intrinsic techniques, which are things like genetics, trace elements, things that are inherent about a bird. And I'm not going to go into a whole lot of detail tonight about each of these different techniques, but I'll just say that all of them have pros and cons associated with them. And so depending on the question that you're asking, you may want to use one over the other or use multiple of these techniques, depending on what your question is. But for this talk today, we're going to talk about how we can use stable isotope analysis of feathers to learn about the natal origins of raptors during fall migration. So first of all, we have to do a bit of a chemistry lesson. I love this part. I was a biochemistry major in college, and so this is where I thrive. So what is a stable isotope? So an isotope is a variation or kind of a flavor of an element that contains a different number of neutrons. So this black sphere in the middle here. And stable refers to the fact that it doesn't undergo any decay and it doesn't change over time. So the element hydrogen has two stable isotopes. Protium, which is here at the top, which has no neutrons in its nucleus, and deuterium, which is here at the bottom, which has one neutron in its nucleus. So this red sphere is a proton, and protons and neutrons have essentially the same mass. And so deuterium is twice the weight of protium, which gives it these different properties and causes it to act differently in the environment. So I like to think of this as in carrying something heavy versus light. So if you were to imagine carrying a light box versus a heavy bag,
Trying to explain heavy
find that the light box would require less energy for you to pick up.
I guess when you go further north.
And I think I'm hearing a little feedback if somebody is unmuted. Thank you, sweet. So if you're carrying a light box, it's going to require less energy and you're going to be able to move more quickly. Whereas if you're trying to pick up something heavy, you might sweat, you're going to use a lot of energy, and you're probably not going to move as quickly. And essentially, this is how isotopes work. Light isotopes require less energy to move and interact. And so in the Northern Hemisphere, in terms of hydrogen isotopes, there's quote unquote more of the light isotope further north. And the heavy isotopes require more energy to move or interact. And there's less of them further north. So we can visualize these isotopes across a landscape because of the differences in their mass and things like environmental temperature, climate, and elevation. The ratio of these two isotopes changes throughout the environment. And so we can visualize the ratio in a map that we call an isotopic landscape, or an isoscape for short. So this is an example of an isoscape for precipitation across the world. And each of these colors represents a different ratio of light to heavy isotopes. So like I said before, there's going to be less of the heavy isotopes further north in the northern hemisphere and more of the light isotopes. So how does this relate to movement and ecology? Well, this kind of relies on this principle of you are what you eat. So the hydrogen from precipitation, which is the main source of hydrogen for organisms, gets incorporated into animal tissue through food and water. So since hydrogen isotopes vary across the landscape, so too will the hydrogen isotopes in animal tissue. And this also applies to us as humans. So our isotopic signatures, if we were to take, a sample of our hair, that would vary depending on where we live in the spatial.
Oh my God.
So since hydrogen isotopes vary across the landscape, we get this hydrogen isotope signal in animal tissue. But the hydrogen isotopes are not going to be exactly a 1-to-1 ratio between precipitation and the tissue due to something called fractionation. So let's say we have some precipitation and maybe it has a 1-to-1 ratio of light to heavy isotopes. This wouldn't actually occur probably in the environment, but this is a good example. And then we incorporate this into an animal tissue, like a feather. And now we have a 3-to-1 ratio. So the ratio changed, and this is due to what's called fractionation. So before we can use this to study animals, we have to understand the relationship between precipitation and feathers. And this technique has been used for a variety of different animals, so things like bats and monarch butterflies, and has also been used in fields like forensics, which I think is a really interesting application. All right, so just to make sure that we're all kind of understanding this, we'll take it one step further. So let's say that we have a juvenile raptor that's growing a feather on the breeding grounds. That feather is going to incorporate the isotopic signature of the environment, taking into account that fractionation. And then when the raptor migrates, it's going to retain that signal of where the feather was grown. And if we collect a feather during migration, when we are handling these birds during the banding process, we can kind of trace that bird back to a general location of where the feather was grown. So for my research, I collected feather samples during the fall migration season of 2020. And this season went on from about mid-August till the end of November. And I collected feathers from juvenile raptors since we could be fairly confident that those feathers were grown on or near the breeding grounds. And these are the 3 species that we collected from: sharp-shinned hawks, Northern Saw-whet Owls, and red-tailed hawks, with the number of individuals that we collected from. And then we chose a subset of those feathers across the season and sent them to a stable isotope facility where they were cleaned and cut and weighed into tiny little capsules and then analyzed alongside some reference materials. And then what came back to us was these values reported in delta notation, which is this funny delta 2H symbol. So whenever you see that, that's just referring to the isotope ratio. So we chose these species for a variety of different reasons. Sharp-shinned hawks are the most commonly banded diurnal raptor at Hawk Ridge. And because of that, we knew that we would have a good sample size across the entire season. We also already know some things about their migration patterns. So we know that females of the same age tend to migrate before males, and that juveniles tend to migrate before adults. They're bird specialists, meaning that they eat mostly other birds. And since they're eating at this kind of high trophic level, they may be more susceptible to biomagnification. And kind of lastly, maybe most importantly, count sites across the US have noticed declines in this species, and the cause of it is not very well known. So this is a species of interest right now for us. The Northern Saw-whet Owl is the most commonly banded nocturnal raptor at Hawk Ridge. They are eruptive, meaning that their numbers fluctuate greatly from year to year, and they're thought to also be mostly nomadic, meaning that their migration patterns might not have consistency from year to year. However, there's really not a lot known about their migration or their breeding range. They're very small and very secretive. And then lastly, red-tailed hawks are one of the most widespread raptors in North America. And there are a variety of subspecies that have different morphological characteristics, including size and plumage. However, the ranges and relationships between the subspecies are not well known. And even within a subspecies, individuals may have lighter or darker plumage. So this is another species of interest at Hawk Ridge. We do have some information about each of these species from band returns. So when we place a band on a bird's leg, it may be banded elsewhere or recovered elsewhere. So each of these dots represents an encounter of a bird that had been at one point banded or re-encountered at Hawk Ridge. So you can see that we do get a large number of returns on our birds. However, in my case, I was interested in where the birds were breeding, which is generally north of Duluth. So if we just look at that area, the number of band returns diminishes greatly. So we don't have a whole lot of information about where these birds are coming from, but we do have some. All right, so that brings me to my first main objective, which was to understand natal origins for these three species. So when we got the data back from the lab, the first thing was just to visualize it. So really the only reason why I'm showing you this box plot right now is to just orient you with how this data appears. So on the x-axis, we have the species, and then we have our delta values on the y-axis. And isotope ratios are just not the most intuitive numbers to look at. So what you need to know is that a more negative number kind of corresponds to a more northern location, and a less negative number corresponds to a more southern location. So from kind of just looking at this, we can see that we had a pretty good spread in our data. So meaning individuals coming from a variety of locations, with some kind of clumps around the kind of average location that a bird might be coming from. We also can see that, for instance, red-tailed hawks have maybe some more individuals that are coming from further north in these more negative areas. So the real thing that we wanted to do here was to visualize where these natal origins were. So it's one thing to look at those numbers, but what do those numbers actually mean in terms of geography? So to do that, created some maps, some isoscapes based on precipitation, created maps for each individual, and then combined those maps together to create maps for each species. So I'm going to walk through each of these individually. So if it doesn't make a whole lot of sense at the moment, that's okay. So to make, first of all, this relationship, remember the fractionation that occurs, we need to know the relationship between precipitation and feathers. I took some data that's readily available from precipitation and paired it with samples from birds that were captured at known breeding locations. So each of these red dots here represents a bird on its breeding ground. You can get a measurement from its feather and then pair that with the measurement from precipitation. And when we do that, we get some sort of relationship like this that we can then use with an unknown origin raptor to figure out where it may have come from geographically. And so from that relationship, we get another map. And this is just a map like our precipitation isoscape that shows the value that would be present for a feather in that exact location. So a purple section in here would correspond to maybe a orangey pink section over on this precipitation map. So then we wanted to actually create maps of where those unknown birds were coming from. And so to do that, we took our feather isoscape that we created from precipitation and known origin individuals. And then we took those individuals from the 2020 season with their unknown origin and used some statistics, which I'm not going to get into the details of that, but I'm happy to talk with you later if you want to hear more about that. And created these probability density surfaces. So most importantly for you to understand is that the parts of the map that are lighter or yellow are higher probability areas of origin for an individual, whereas the darker, bluer regions are lower probability. So high probability and low probability. So we did this for all 240 individuals, and I'm not gonna bore you tonight by showing you 240 maps, but I do want to show you just two examples that show the potential for this technique to tell us something. So the first is from Sharp-shinned Hawk. We had two individuals that had significantly lower isotope values than the other Sharp-shinned hawks. And what this corresponded to in the map was a probability density that was highest in regions of northern Canada, so up in here and into Alaska. So if we remember back to the band returns, we only have one record in all of our band returns of a Sharp-shinned hawk migrating from that distance. And so this raises, some questions. Is this migration more common than our banding records suggest? Or, what is it that caused these Sharp-shinned hawks to migrate in that path versus, coming down here in this Rocky Mountain region? The second example comes from the red-tailed hawk. So this was in collaboration with another researcher, Ali Pisano, who I believe is on the call. And she was studying specifically dark morph red-tailed hawks. So if you remember, I told you red-tailed hawks have many different subspecies, and they can also have different color morphs. So two of the individuals that I analyzed were dark morph individuals like the one shown in this picture. And this hawk in particular had some plumage characteristics that suggested that it belonged to the Harlani subspecies. And so if we look at this map on the left here, the suspected breeding range for Harlani is shown here in purple. which extends down from Alaska into the Yukon, and even down into kind of British Columbia and Alberta. And then if we look at the isotope assignment, we had the highest probability up here in the Yukon, which is definitely within the Harlani range. So this kind of acted as almost a positive control, and is kind of a cool result to get. The other individual that was also dark morph had a high probability of being not quite as far up as this individual, but still up in Western Northern Canada. So to kind of simplify all of those maps and to view them more easily, we took all of those individual probabilities and combined them together for each species. So the maps that we're going to look at next show the probability that any individual came from a cell in the assignment area. And this is going to be on a species by species basis. So in this example, the green area indicates the highest probability for that species, whereas the lighter or the pink color is a lower probability. So this is what the maps look like. So again, the darker regions represent higher probability density than the lighter regions. And each of these maps is constrained with known breeding ranges. So this helps to eliminate regions and habitats that these birds were unlikely to be found in. So for sharp-shinned hawks, this created a probability density that showed the highest probability regions for an individual in kind of northern Minnesota, up into Ontario, Manitoba, and Saskatchewan, with kind of a decreasing density the further that you go north. For saw-whets, we have this kind of large region of high probability densities that spans kind of across the US. We have, if you remember back, I guess I didn't point this out, but we have band returns that span almost the entire length of the US. From over in British Columbia, we have 2 band returns, which I think is just incredible for a saw-whet owl to, move those kinds of distances. So it's kind of hard to eliminate any of these regions in here, but probably the majority of the birds that we banned are coming from this kind of region up into Canada. And then red-tailed hawks showed kind of similar probability densities to sharp-shinned hawks. However, they showed a higher probability further north up into Western Canada. So one thing that, maybe you're thinking or might be kind of obvious is that there's some areas on these maps that we would not suspect that our birds are migrating from. So we would not necessarily expect birds to be coming from this Rocky Mountain region range for these individuals. That's a long and difficult migration. And the only reason why these areas are showing up as kind of high probability areas is that their isotope values are similar between this location over in the western part of the United States and this location up here in southern Canada. So this is kind of a limitation. So just kind of to summarize that, we saw mainly the highest probability densities up into northern Minnesota and into Canada. However, some individuals are definitely coming from much further north than that. And we also see that assignments using hydrogen isotope analysis alone are quite broad. and we'll come back to that. Okay, so moving on to the second objective, which was taking those locations that we've now kind of identified and determining whether or not there are migratory patterns within them. So birds can exhibit different strategies, like I already mentioned. Our question was, how does the banding data at Hawk Ridge relate, the banding date at Hawk Ridge relate to natal origin? So since hydrogen isotopes have this general latitudinal north-south trend, we used it as a proxy for latitude. And we created a relationship between isotope value and banding date, and then looked for trends. So our kind of predictions of what we might see were that, one, we could see this trend going down. So let me orient you to this first. So we have banding date, so beginning of the season till end of the season, and we have isotope value. So remember, closer to down here is more north, and closer up here is more south. So this downward trend would indicate that maybe southern populations were banded earlier in the season than more northern populations. We could see the opposite of that, with more northern populations banded before southern populations, or we could see no pattern at all. So we kind of predicted that both sharp-shinned hawk and red-tailed hawk would have more southern populations banded first. And this is due to patterns that are observed in other parts of North America and what we can kind of observe also morphologically of these species. But for Northern Saw-whet owls, we didn't really expect to necessarily see a significant trend due to their somewhat nomadic behavior. So the graphs that we're about to look at are going to be just the same as this. It's going to have banding date on the x-axis and then the isotope values. And we'll look to see whether there's this downward trend, upward trend, or no trend at all. So let's take a look at sharp-shinned hawks first. So first of all, we've separated these out by sex. So in the red circle, we have females. In the blue square, we have males. So sharp-shinned hawk females, their isotope values had a significant negative trend with banding date, which indicates that expected southern individuals coming through Duluth or being banded at Hawk Ridge sooner in the season. However, while we saw a trend for males, that trend was not significant, which was interesting. And I'll come back to that. For red-tailed hawks, we also saw the expected trend of potentially more southern breeding individuals migrating through Duluth earlier in the season versus more northern individuals. And we did not have sexes for these individuals, so we weren't able to look at trends there. And then Northern Saw-whet owls were definitely a bit of a surprise for me. Females showed a significant positive relationship with banding date, suggestive of northern breeding individuals being banded earlier in the season than southern breeding individuals. Just like sharp-shinned hawks, though, males showed a similar trend, but it was not significant, which is a kind of interesting result. So just to briefly summarize that, we found that migratory patterns depended on species and sex, that sharp-shinned hawks and red-tailed hawks exhibited kind of the expected patterns. However, males of both sharp-shinned hawk and northern saw-whet owls did not exhibit that significant migration pattern, suggesting that perhaps they're doing something else before making their migration through, and that there might be some sort of, mixing during that pre-migration time. And then in addition, we saw kind of an interesting trend with northern saw-whet owl females that could be showing that more northern individuals are banded earlier, or could potentially have to do with some of their east-west movements. So to give kind of a broad conclusion to all of that, hydrogen isotope analysis definitely has potential to help us understand natal origins and also patterns within migration. And we found that there was, in some cases, a significant relationship between the isotope value and the date that that individual was banded at Hawk Ridge. So just to quickly put this into perspective, why is this all important? Well, for migratory species that use many different locations during a whole year, it's really important to be able to connect those locations in order to have effective conservation and management. A negative impact on any of these locations, such as habitat loss, has the potential to, affect survival. And with things like the avian influenza, it's really going to be quite important to know how birds are moving and mixing. So with, kind of the knowledge of this, we can use that to be better prepared to conserve species into the future. However, I have, suggested some limitations to this research. So there's definitely some ways that it could be improved. So one of those is using a dual isotope approach. So researchers have recently published a paper that used a combination of both hydrogen and strontium isotopes, and they found an order of magnitude increase in the assignment precision. So these maps over here on the right show an assignment based just on hydrogen isotope, just on strontium, and then the combination of those two together. So if we have some complementary isotopes, they can be combined to both create a latitudinal and more of a longitudinal gradient. Another area that I think would be interesting to look into is looking at a multi-year analysis. So all of this data came from 2020, and perhaps these trends are not apparent in other years, and that would be interesting to look into. Another area that I'd like to pursue is looking at combining isotope data with other variables that we collect during migration. So this is things that I'm referring to here are things like contaminant data. So can we put data from contaminants on birds into a geographical perspective using isotope data? And then finally, I think combining isotope data with other techniques is really the most fruitful way to use isotopes. And so one of those obvious techniques is using transmitters. So I just wanted to highlight one really cool thing that happens this past fall. Hannah Tutangi helped to deploy this first cellular transmitter on a northern saw-whet owl at Hawk Ridge. So transmitters are becoming available for smaller species, and it is absolutely incredible. And we have our friends at Cellular Tracking Technologies to thank for this. There was also a transmitter put out this fall, not at Hawk Ridge, but on a sharp-shinned hawk. So the possibilities for getting really high resolution data and, combining that with isotopes is definitely there. So just some takeaways. Definitely think that stable isotope analysis can be a useful tool to study migration, depending on what your questions may be. Definitely has the potential to be paired with other data and may be useful for studying how migration shifts over time and from year to year. So just to end today, I'd like to thank all of the people who made this work possible. My advisor, Matt Ederson, Hawk Ridge Bird Observatory, specifically Frank Nicoletti, who without his help, we wouldn't have any of these birds to study. And I'd definitely like to thank my funding organizations, the Minnesota Ornithologists Union, Hawk Migration Association of North America, and Sigma Xi grants in aid of research. And I think with that, I'll answer any questions that you might have. So thank you for listening, and thanks for spending your Tuesday to learn about birds.
All right, looks like we have a question in the chat. Let's see, in reference to constrained breeding range averages, are your probabilities for sharp-shins and saw-whets higher than those for red-tailed hawks since the values are lower? If yes, why do you think there may be differences in the probabilities between the species?
I think I understand. Let me take a look at the question just
Yeah.
to make sure I understand. So for those maps, so I didn't talk about what the values mean. So the values are probabilities, probability densities, which means that if we were to add up all of those probabilities, it would equal 1. And so the reason why they are different is because the projection on which it was done are different sizes, if that makes sense. So the red-tailed hawk versus the Northern Saw-whet Owl and the Sharp-shinned Hawk all had different maps that they were projected on. So that's what caused those numbers. Yeah, the numbers are not super useful to look at, just because they are these kind of probability densities, which is not like a very meaningful value, if that makes sense.
Okay. Yeah. Yeah. Gotta thank you. Do we have other questions from folks?
Oh, Bob said, where do you hope your research will take you next? That's a good question. Well, I'm happy to still be in Duluth and definitely volunteering at Hawk Ridge. And I think we might have some interesting upcoming research that maybe I'll talk about next year that might actually, combine some stable isotope and transmitter work. So look forward to that. Yeah,
I was curious if we had any updates. Did the legislature do what they were supposed to do?
Oh, they're still working on stuff.
Oh, okay.
Yeah. Allison asked, is there anyone else doing similar work? Yeah, so that's a good question. So there definitely are people using stable isotopes for many different applications and also for birds. I try to at least make it somewhat clear that there are definitely limitations to this. And it's hard to, I think, as a researcher, want to use something that doesn't have as much resolution as something like a transmitter, and with transmitters becoming so much more readily available, I think stable isotope analysis has become maybe slightly less popular, but I think that if combined in the right way, they can tell you different information. And so I haven't really run across a lot of people who are combining both of them currently. And there isn't currently a lot of work in the raptor world besides that paper that I cited that are working on isotopes in this context.
Cool. It's fascinating to think that you can use feathers to say, okay, this is where you were born, or likely where you were born. just amazing.
It is pretty incredible. And stable isotope ecology is like a huge field that's used for so many other things as well, which also fascinating. And I definitely don't have as good of a grasp on, but you can learn a lot by looking at the chemicals in an organism.
Yeah, well, we use feathers for birds for testing for so many different things. It's just amazing what they can tell us as a whole. Anybody else got questions for us?
Yeah, could I ask a question?
Sure.
Yeah. Hi, Dick.
Hi. Your pictures of the locations, returns for bandy birds for sharp-shinned hawks, looks like there's sort of a line going south from Duluth. So are these birds caught at different times?
Yes. Yes.
So would it be possible to say sort of where they are? Sort of have a map where, I mean, I assume that they're not going to the places that they're found, right? They're going through there.
Yeah. Yep. So what you're saying is by looking at those dates and comparing them to the locations, you could kind of trace that migration
Right.
temporally. Yeah, I definitely think that, yeah, that should work.
But it's not like if you're looking at range maps and you say, okay, these are where they spend the winter, but where you're catching the birds is not where they spend the winter. Right.
Right. Yeah. Yeah. No, I think that's a great point. And I think it would be interesting to look at that band return data in that way because I think you could learn a lot just by, looking into that data set a lot more. I think there's a lot to tell us.
Good.
All righty. Anybody else? Well, thanks so much for coming, folks. I am going to, let's see. Emily, why don't you make me the host again?
I can do that. Stop sharing.
Yeah, and I just wanted to leave you with a parting thought here. Hawk Ridge is a nonprofit, and out there in the real world is where we get the money to do the things that we do, both for education and research. And we strongly encourage anybody that wants to help us continue doing research and work like that to stop in on our website, become a member of Hawk Ridge. That's a great way to support what we're doing. You can donate money specifically designated for different kinds of projects, any of those kinds of things. And I'm really, really appreciative to all of the folks out there that I know do that on a regular basis. So I'm just putting that out there, the normal nonprofit spiel. It's really important. And I've learned in the course of my career that people appreciate hearing that rather than dread hearing it. So I'm just going to put that out there. Thanks so much, everybody. The plan is to take this recording and we will put it up on our website, on our YouTube website. So I'm going to stop the recording now. But if anybody's got any other things that they wanted to